Hi folks, welcome to class 15. This is the last video I'm going to be recording before I get to talk to you guys. So if there's anything that I'm doing that's totally terrible, with luck, we'll find out in the synchronous question and answer session tomorrow. In the meantime, hope things are going well, you're staying safe, you have coffee, all the important stuff. Not sure how many of you will recognize the picture on screen. This is from South Park. If you haven't ever watched South Park, it's highly sweary. It's seriously inappropriate for any audience, and it's really funny. So this is, one of the teachers in the South Park, and drugs are bad. Okay, don't do drugs. Was one of his messages he used to tell all his students. We'll be doing drugs, or at least talking about drugs, later on today, and then probably for the next class as well, when we talk about addiction and so on. This is today's, pretty neuroscience picture. I'm going to do this every class. These are stem cells. So the purple in the center here is the reservoir of stem cells in the brain. It's stored or exists in the periventricular zone, right around the ventricles. And as we mentioned briefly in class at one point, we know now that mammals, including humans, do have adult neurogenesis. They have lifelong production of at least a small number of new neurons. And in fact, those new neurons turn out to be really important for things like resolving depression, as we talked about, but also for making new memories, supporting cognitive function. It's a big deal, the fact that we have these new neurons. And so what you're seeing here, the green stain, is as the cells are differentiating into neurons. That's a marker for neuronal cell types. You can see it doesn't exist in the pre-neuronal stem cell mass, but as these cells move out and move to where they're going to be in their adult lives, they turn into neurons, start expressing different proteins, and you can mark for that. Reminder on this, I'm going to try and touch base with you individually probably, but just if at all possible, I'd really like to have annotated video presentations of your paper reviews. If that's truly not possible, again, we'll talk about this a little more tomorrow, then okay, fine, we'll live with that. But ideally, if you can get hold of, a Zoom session and the person with whom you're presenting or people with whom you're presenting, you should be able to record that session, and each one of you should be able to speak as part of that recording. I believe that should be fine. Anyway, try it, let me know how it works. Any advice you have for your colleagues would be really great. Thanks. Last time's paper, we talked about the Tong et al. Paper looking at the release of GABA from these AGRP neurons. I like this, as I mentioned, both for the methods and for the really clear rationale. The abstract takes you through it in simple, easy steps to understand. These neurons are important. They release three things. If we delete two of those things from the animal, nothing happens. So it's probably the third thing. It's probably GABA. But there's a real problem. We can't just delete GABA from the animal because that'll cause it to have seizures. And so, the Krelock's methods, where they take out this vesicular GABA transporter, this protein that's responsible for putting GABA into vesicles at the end of the neuron to allow them to be GABAergic neurons, to allow them to release GABA. They put this under control of the Krelock system, so it can be taken out, and you end up with this AGRP set of neurons. Those cells only have the VGAT removed. And the reason that works is that Cre is put under the control of this AGRP promoter. So this cell type specific promoter. In cells then, in the animals that have both these parents, we talked about that last time, a Cre parent and a LOXP parent. In cells where Cre is active, so in cells where that reporter gene activator is taking place, in this case, for this paper, it's the AGRP promoter. So in AGRP cells, the gene gets cut out, and in some cases, like I mentioned, you have a reporter gene that gets expressed instead. So for instance, a green fluorescent protein saying, yeah, it worked, technology is happy, these cells are fluorescent. So in your offspring mouse, one parent has Cre, one parent has the LOXP sites. These are both completely wild type, nothing is happening in them. And in most of the offspring mouse, most cells don't have Cre turned on because the promoter isn't active. So most cells, again, nothing happens. The target gene is perfectly intact. Only in a few cells, in the case of this paper, the AGRP cells, is the gene taken out. Because the gene is a GABA gene, the only cells where something really functionally is happening are cells that are both AGRP cells, so Cre is turned on, and they're GABA cells, so it matters whether they have VGAT. And so the authors of this paper were able to target very specifically AGRP GABA neurons, this very small set of cells. We spent a bunch of time last time talking about the kind of twin concepts of homeostasis and adaptation. The idea that your body and your brain have set points they're trying to maintain for things like temperature, fluid volume. In the case of this, RH is recurrent hypoglycemia. So your brain's trying to maintain fuel supply, but also it adapts. And so if you are repeatedly hypoglycemic, if you are an individual, usually a type 1 diabetic, that's taking repeated doses of insulin, becoming hypoglycemic again and again, what your brain does is say, I guess that's the new normal. Kind of like we're all doing this by remote for COVID-19. It's the new normal. And so the brain adapts. The brain says, okay, well, if I'm going to be in this condition of repeatedly low blood sugar, I'd better adapt. I'd better make a whole bunch more glucose transporters so that I, the brain, still get the glucose I need. And because of that, you get smarter, but it means you can't detect subsequent episodes of hypoglycemia because the brain is protected from that, because the brain is now much more able to get glucose when it's needed and doesn't have it as good an ability now to detect when the animal is hypoglycemic. As I mentioned, the reason we didn't have Dr. McNay's insulin clinic is because other examples of that adaptation include storing weight, not expending energy. Here's the expenditure. This is the motor activity. Your usual rat, pretty active, less during the day, more at night. Your recurrent hypo rat, still more active at night, but much less active the whole time, burning fewer calories because the brain says, the new normal is I'm repeatedly starving. I'd better turn down my energy expenditure. I'd better conserve my fuel reserves. I'd better basically get fat. We talked a bunch about non-human cells, microbes, and other things that can make you fat. As I mentioned, we'll talk more about non-human cells, parasites, and so on when we come to talk more about consciousness in the self. But certainly there are good data now that one factor that plays a major role in creating your phenotype, obese versus lean, is the makeup of your gut microbiome. So some microbes are very good at getting energy to be absorbed into the body, tends to make you fatter. Some microbes, much worse, tends to make you leaner. And as well as cells outside your own DNA, if you like, being in control of some things, different parts of your brain are in control and are at war with one another, in conflict. And in particular, you've got this prefrontal cortex and frontal lobe essentially watching the rest of you, acting as a brake. And in some cases, particularly in the case of eating disorders, if it's overactive, it's a negative brake effect and it will stop you from eating appropriately. It'll tend to tilt you towards the long-term, no, you shouldn't eat that, you might get fat, stop doing that, as opposed to the healthy, regular balance of, hey, that pizza looks tasty, let's go eat it, let's have normal weight. This concept of self-control is one that has a bunch of different ways to think about it. So we can think about it as the frontal lobe acting as a brake. We can think about it as being, in some ways, your conscious self, making sure that your primitive lizard brain doesn't do what it wants to do, doesn't hit people, doesn't smash things when you get mad, doesn't, in some cases, sure, have 75 cookies because they taste good. And we talked about the fact that just like other processes, self-control, almost certainly requires glucose. Everything your brain does requires glucose. There's no reason why this should be different. It's this negative GABAergic, in fact, influence in the rest of the brain. And in fact, GABA neurons do require a lot of energy. They fire quite fast. And so it takes a bunch of fuel. And we talked about the concept of the Stroop test as depleting that self-control. So if you're told to name the colors these words are printed in, or even just to read the words and pay no attention to the color, but the first is harder. If you're told you have to say red, blue, green, yellow, green, black, and actively suppress reading the words, which is the default, that's self-control. That suppression of reading words requires energy, requires brain power, depletes a resource. I couldn't find the video, I tried to find it after the last class. There's a couple of good videos for things on screen where words pop up in random places, and you're instructed to actively not read the words, to actively not think about pink elephants, to prevent yourself from doing a natural default behavior. This is now a very controversial field as to what's going on. So Roy Baumeister is the head of the lab where much of this first work was done. He's generally responded pretty well to multiple failures of other people to replicate that work. It's not helped by the fact that the first author, a guy called Matt Geyer, is a complete twit and really doesn't understand the brain, really doesn't understand glucose. He came to give a talk at one point, and this is my research field. He thought glucose was a protein, just really quite poor. This is a recent review paper, and if you're interested, I think it does a decent job, because we wrote it, about discussing the issues. And the basic conclusion is, yeah, self-control really does require glucose. It's not special. It doesn't require more glucose than anything else, but just like all other cognitive processes, the experiments about ego depletion and self-control. The basic concept here I mentioned briefly last time. If you have a really complex number to keep in your memory, for instance, you're more likely to fail in self-control, give in and have a cookie if one is offered to you. Similarly, if you're told to not pay attention to words and actively suppress paying attention to words on the screen, you're more likely to make errors in the Stroop test. And so here's that graph from last time again. If you just watch the previous video without any particular instructions, then it really doesn't matter whether you're given sugar or Splenda while you do the Stroop test. You're pretty able to do the Stroop test normally. You have enough glucose, you haven't depleted anything. The gray bars are where you were previously told to watch a video and actively exercise self-control, actively not read words that pop up on the screen. And then if you're not given any more extra sugar, you tend to do worse at the Stroop test. You tend to make more errors. The concept is you basically used up that self-control reserve. And that self-control probably is, like all other cognitive processes, like we saw in the maze task with microdialysis. You deplete glucose when you think hard. If you give a little extra sugar, just like you would make a rat smarter running around a maze, you also improve the ability to do the Stroop task. You now make fewer errors on that task again. As we were thinking about various things affecting, brain glucose and eating and so on, we talked about anorexia. And again, the fact this is in some ways a disease of over self-control, of exerting too much of a top-down influence, causing all kinds of negative symptoms. You misestimate your own body mass, you have inappropriate ideas of what is attractive. And what are the reasons for this? One of the reasons, has to do with that dopamine binding. We saw previously that in obese subjects, you maybe get too little dopamine binding from a donut, so you eat more than one donut. In an anorexic subject, it's quite possible you have too much dopamine binding. And so one reason you might be susceptible to anorexia is you have an abnormally high level of pleasure from a small amount of food. The second reason is shown here. This is the balance between your frontal cortex and your amygdala limbic system. When you see a piece of cake, your limbic system generally says, hey, looks good, tasty, I should eat cake, positive approach, go towards the food, eat the food, tilted towards the short term. At least for the first piece of cake. Maybe the third piece of cake, your frontal lobe kicks in. In the anorexic brain over here on the right, that balance of initial responses in your brain is out of whack. You have too much of this top-down GABA negative influence from the prefrontal cortex. You have an over self-control. And that causes you to avoid the cake. You'll stay overly thin, and you'll also set up this negative association. When you saw this cake, your overall response was to feel bad and avoid it. That's going to create a memory. We'll talk about memory later in the semester, but you're going to make an association between seeing high-calorie food and feeling bad. That will generally cause you to avoid that food even more in the future, which is why you get this negative spiral down into anorexia. As usual, this is the you're not really here, you can't ask questions slide. Almost certainly we'll have a compulsory question session on Blackboard soon. I know we've talked about having questions in the chat. We'll definitely have a compulsory question session, probably after the first 1 or 2 synchronous class times. So the next topic I mentioned, I always want to talk about this, we're going to do drugs, and I always hear myself, and that It's not quite what I want to say. We're going to talk about drugs. We're going to talk about what happens in addiction, why certain substances and behaviors make you feel so good, give you a high. This is a great book, Elephant and Acid. There are at least 2 reports in the scientific literature of elephants being addicted to various substances, one of which was done by the CIA. Who knows why they got funding for that. But more commonly, in fact, elephants are used in Southeast Asia as ways to smuggle drugs through the jungle. And so one of the ways you can control your elephant is to get it hooked, is to make sure that you are the person that is supplying your elephant with heroin or cocaine or whatever. There are a couple of instances where these elephants have been put into rehab. And you can imagine that an elephant going through withdrawal and getting pretty unhappy about the experience needs a whole bunch of methadone or whatever, and is really quite hard to restrain. So the experience of addiction is not unique to humans. You can certainly make lab rats addicted to various substances. What you are actually addicted to is dopamine, right? This is the small molecule in a particular part of your brain that gives you that spike of pleasure. It gives you that high that creates the association between, hey, I took this white powder and I snorted it up my nose, and man, that felt good. I should go do it again. Creates those kind of cravings. And so the particular place where that dopamine is important is in this medial forebrain bundle. So labeled here as MFB, medial forebrain bundle. It's this single pathway between the nucleus accumbens and the ventral tegmental area. And that's commonly referred to as the brain's pleasure center. It's really a single path of dopamine neurons. And that's where you get dopamine release when you are having addictive responses, when you're taking something that gives you that pleasure. The way that different substances work, crack is no exception, cocaine, anything that makes you happy releases dopamine in this pathway. The reason that some substances and drugs are addictive is because they give you this really rapid rush, a very high level of dopamine release, very rapidly produced, causing intense pleasure, obviously associated with a particular substance. You're very good at remembering, oh yeah, I took this stuff and made me feel good, I should go get some more. And so you rapidly create that very strong association that causes you to want to go do it again. And that's addiction. Notice that not all dopamine neurons are these pleasure neurons, right? So for instance, we have dopamine neurons in the substantia nigra, part of the basal ganglia. Those are the neurons involved in Parkinson's disease. They're involved in motor control. They don't give you pleasure. That's an entirely different set of dopamine neurons. The ones we're talking about is this mesocortical limbic pathway from the nucleus accumbens, causes dopamine release in this one particular set of neurons. And you can measure that, right? If you measure the level of dopamine being released in that pathway, you can actually determine, yes, this is exactly how much of a high the animal or the human is getting. And you can do that with an electrical impulse. You can put an electrode into that pathway. And if you do that and give a rat access to stimulate that pathway, the rat will continuously push that lever or that button, stimulate this medial forebrain bundle, and will actually do so to the point of death. They will ignore food, they will ignore sex, they will ignore water. They will just stimulate themselves over and over and over again, constantly getting this dopamine rush until they die. So it's an extremely powerful driving force. The reason that it's very hard to break addictions is because we are set up, our brain is wired to want dopamine. It's really very strong. Incidentally, I do kind of wonder sometimes why there isn't almost a market in black market brain surgery. Rather than having to go out and constantly buy dubious packets of white powder from shady guys on street corners, just get your friendly local brain surgeon to give you a button and an electrode into this pathway, and you're set, right? That's your addiction taken care of forever. Forever won't be very long, but it seems like it would work well. Here's the experimental setup where we can show that it's dopamine. So here's the electrode going into this medial forebrain bundle. And what you see is that you can measure dopamine. If you give a dopamine blocker into this brain region, you prevent the addiction. You wipe out the pleasure. You avoid the animal becoming hooked. So as I mentioned, animals will happily self-administer either electrical stimulus, or they'll give themselves cocaine. If you hook an animal up to a little cannula of cocaine, it will again happily press that lever, keep overdosing itself, often to the point of death. If you take out those brain regions, if you lesion, the nucleus accumbens, the mental tegmental area, you no longer get that association. Dopamine doesn't get to have its effects. And so you can't get addicted, basically. The animal won't show addictive behavior. Hasn't been tried in humans, not particularly ethical, but I am sure the same thing will be true there. Notice the mention on this slide, natural reinforcers. We're not set up to have, opioid receptors in the brain just so we can get hooked on heroin and morphine. Those receptors exist because there is an endogenous ligand, an endogenous substance that binds to them. In this case, endorphins and enkephalins. Your brain produces several substances that give you the feeling of being high, that reduce pain, and so on. We'll talk about that, in context of runner's high in a second. And so those receptors are hijacked by things like cocaine, heroin, other addictive drugs, use our body's endogenous systems to give us that dopamine release. The drugs work in various different ways, and we're not going to go through them all. Here's one example, though. So cocaine, right? Cocaine stimulates dopamine release, and it also blocks dopamine reuptake. So you'll remember from the discussion of Prozac and the discussion of various synapses, once a neurotransmitter, in this case dopamine, has been released into the synaptic cleft, it binds to receptors and then rapidly gets either broken down or taken back up. And so for dopamine, there are reuptake transporters into the presynaptic cell. One thing that cocaine does, as well as stimulating that release, is to block that reuptake. So it's causing a rapid dopamine release, and it's causing it to hang out in the synapse for longer. Overall, you get a very intense burst of dopamine stimulation. You see the receiving neuron here. All those dopamine receptors are active, and they're going to be kept active for a little longer because the cocaine is preventing the removal of dopamine from the synapse. I mentioned other drugs act slightly differently, but the general endpoint is always a bunch of dopamine hanging out in the synapse, causing intense pleasure. As I said, it's not just substances to which you can become addicted. If I am in a period when I'm actually doing some decent training, the half marathon I signed up for has been canceled, but one of the few benefits of being cooped up here at home is I actually have more time to go running. It is definitely true that you get what's called a runner's high. If you run for a while or you work out intensely, you do something that places a reasonable degree of physical stress on your body will respond by releasing endorphins. These are, as the name suggests, Endorphin means endogenous, coming from within, and they are substances that activate the body's own opioid reward system. So is it possible to get addicted to running? Absolutely. It's a thing you do that causes you to release a pleasurable stimulus within that mental tegmental pathway. You can get hooked on running. And certainly if I've been exercising several days, a couple of weeks maybe, and then I don't run for a couple of days, I get particularly cranky. Basically, I'm having withdrawal from running. So you can get addicted to anything that gives you pleasure, whether it's nicotine or binge watching Netflix or running. Some of these addictions are more or less harmful than others, but they all have the same biochemical basis. They're all an activity that gives you an intense chemical pleasure and that you can get hooked on, that you want to go seek out over and over again. Many things can be pretty deadly, right? But they vary a lot. Running is probably not going to kill you. Cocaine's going to kill you pretty fast. If you give a rat unlimited access to cocaine and they can self-administer, in 25 days, they're all dead. Heroin is not quite as deadly. Different drugs have various different characteristics, how addictive they are, how painful withdrawal is. We'll talk in a little bit about some of these different drugs. The withdrawal from cocaine is relatively mild. The withdrawal from nicotine is horrendously painful. So even though nicotine won't kill you as fast, it's actually harder to kick the habit than it is to kick a cocaine habit. So different drugs, just have different characteristics of things like fatality and danger, even though they all have that same dopamine release. Chocolate, especially British chocolate, I can't imagine getting hooked on Hershey's, but Cadbury's before it got bought out by Kraft, absolutely. Intense pleasure, very tasty, probably not going to kill you, probably will make you fat, right? But it's certainly a thing on which you can get hooked. Why is there a picture of a syringe? things that are associated in your memory with the thing to which you are addicted are likely to trigger cravings. So Cadbury's. Cadbury's comes in a characteristic purple wrapper. If I see that purple wrapper, I definitely am reminded pretty rapidly, oh yeah, chocolate. Chocolate's really good. Now, am I addicted to chocolate? No. Do I have that strong memory from the pleasure that chocolate gives me? Absolutely. It used to be that posters for things like drug therapy clinics would be covered in pictures of piles of white powder and syringes and so on, until we realized that's really dumb. What those pictures are doing are reactivating the memory for the addiction, and in many cases causing the patient to relapse, to go generate cravings and go seek the drug. And so now if you're ever at a drug relapse clinic, you will never see a syringe or a picture of anything that might be thought of to trigger those cravings. I'm talking about addiction here as a memory. That's very much true. If you can't remember the thing to which you're addicted, you can't become addicted because, what are you addicted to? You don't know what to go out and seek. And so addiction has these two components. It has the intense pleasure, and then it has the memory. Oh, what was it that gave me that intense pleasure? Oh yeah, I should go do that again. One of the reasons that marijuana is less addictive is because it has the side effect of impairing your ability to make new memories. Don't smoke marijuana when you're trying to study because you won't remember what you were doing. But one of the reasons why it's harder to get hooked is because your brain is not as good at storing the association between what was it that made me feel good? I don't know. I can't remember. That is not an addiction. You don't know. You don't have that memory component of the addiction. Obviously, drugs do things that aren't just giving pleasure. There are lots of side effects. And the role of learning and memory is true there also, right? So tolerance. Tolerance is the phenomenon where you build up a resistance to a particular drug. So you take a small amount of a drug, you experience a symptom. You are resistant, in many cases, to having those same side effects. How is that created? Well, firstly, you've got to experience the effects, right? You only experience tolerance to things that you see. So if you're having an addiction to alcohol, one thing that alcohol can do in large doses is cause you to have convulsions. If you never drink to that extent, you won't experience any tolerance to that because you didn't experience that symptom. Your body actually has to experience something and adapt, has to say, oh, well, okay, this happened. It happened a second time. I guess this is normal. I will become tolerant to it. And so often you develop this tolerance only by experiencing a particular side effect. Because this again is part of a memory, it is most strongly seen in the same conditions where that memory was formed. So if you are a cocaine addict and you always take your cocaine in the same place, what will actually happen is you get maximum tolerance in that place. And so you'll increase your dose to get the same effects because you've built up maximum tolerance, because the memory is stronger in that one place. Sadly, of course, this can mean that if you go somewhere else and take the same dose, now it's an overdose. Now you can have really negative effects, possibly lethal, because you don't have the same tolerance somewhere else. Likewise, as I mentioned a moment ago, because it's a memory, you can get withdrawal. And so if you go back to that same environment where you used to take cocaine, it is very likely to generate the memory of, oh, it used to feel good, and now not having the drug, and you feel bad. You generate that withdrawal. So learning and memory are a key part of addiction, of tolerance, and we'll go through tolerance a bit more in a second, and of withdrawal. The reason that drug addicts who are trying to kick the habit are told to avoid their old hangout spots, their old friends with whom they used to do drugs, is because all of those are likely to activate a memory system, trigger the memory of how good it felt to take the drug, and much more likely to relapse. You've got to break all those cues to be effective at getting clean, basically. The brain develops tolerance to overall metabolism. Things that are stimulants, like cocaine. So cocaine, you take, if in a highly unethical world, like I came around right now and gave you all a little cocaine and put you in an fMRI, you would see increased brain activity. Your brain would be revved up for a minute. You'd be, it's a stimulant. You'd be very high and hyperactive. Your brain adapts really rapidly to that and lowers overall brain metabolism. So here is the brain of a control subject, and here is the brain of a cocaine addict. Baseline conditions, just talking about maybe soccer or the weather or their pets or something. And you can see there's much less activity in the brain of the cocaine addict. This is adaptation. This is this homeostasis thing again. Your brain says, holy cow, I'm repeatedly getting these really hyperactive episodes where I've got much too much going on. I better tone myself down. And so in the absence of cocaine and the absence of the things which they're adapting, your baseline activity goes down a lot. Stimuli related to that addiction, though, are particularly salient. They are particularly able to cause activation. So here are those same people. Here they are talking not about football or chocolate. Now they're talking about cocaine. And what you can see is that the frontal lobes of the cocaine addict brain massively light up. We're talking about things that are very, very salient to them. Their brain says, oh yeah, cocaine's really important. How can I plan to get some? And so unlike any other stimulus, stimuli related to the drug habit are actually particularly effective at triggering brain activity. One thing that has been called into question a couple of times is there that such thing as an addictive brain type? And the answer is probably yeah. You can think about this in the context of, once again, dopamine binding. Imagine that you were thinking about getting addicted to donuts. Pretty unlikely, as it turns out. You take a donut, you take a bite, you get a small, normal amount of pleasure, not really likely to get addicted. And so here, for instance, is that experiment with nicotine. Here's the amount of nicotine released here's the amount of dopamine released in the brain of a non-smoking population given a nicotine dose. And it's pretty average, pretty normal baseline. In the brain of a smoker, much more dopamine binding in response to a nicotine dose. Basically, an increased amount of pleasure for that stimulus. So back to my donut example. If you took a donut bite and it was the most amazing thing in the world and you were flooded with pleasure and joy and everything felt tingly, you're much more likely to get hooked on donuts. And so the addictive brain personality type, it's not really a personality type, it's a biochemical type, is in fact that you get more dopamine release to that particular stimulus. Think about the experiment with the electrode and the microdialysis. If you put a dopamine blocker in and gave someone a donut, there's no way they're going to get addicted because they're not going to get that pleasure. If your brain is set up to release a very large amount of dopamine in response to donuts, or in response to running, or in response to watching kitten videos, whatever it is, you're much more likely to get addicted to that thing because you form this much stronger pleasure association with that stimulus. And so yes, there is an addictive brain type, and it is a type that is set up to release large amounts of dopamine in response to whatever the stimulus is: cocaine, nicotine, video games, gambling, whatever. Consistent with that, what we see is that people who are addicted to a certain either behavior or substance tend to seek out a fairly constant level of total dopamine release. This again is very similar to what we saw with obesity. If you don't release enough dopamine, you eat more to get that baseline homeostatic level of pleasure. If you're addicted to a drug, and let's say you release a fairly low amount of dopamine, either because your brain was initially set up that way, or because your brain's adapted to repeated overdoses of that drug. So low amounts of dopamine release, you tend to go out and get more drug of substance. So on the x-axis here, how much cocaine did these individuals take each week? And what you can see is that people taking more had less dopamine released by cocaine. So they needed to take more to get that same amount of pleasure. If they're releasing a fairly high amount of dopamine for a fairly small amount of cocaine, that's enough, right? That's got them to the point where they have the pleasure they were seeking. You don't need to take any more. So the idea that we can look at addiction and pleasure seeking as a homeostasis mechanism, as the brain trying to achieve a certain desired amount of pleasure, a certain desired amount of dopamine binding, turns out to have some pretty decent value. Obviously, there are side effects that aren't just brain side effects. Alcohol, for instance. In overdose, it will kill your liver. That's the picture of a cirrhosis liver. You'll be unable to process other toxins. You'll have a whole bunch of nasty side effects. It's not just the brain that's being affected here. One especially nasty example is cocaine. And I have to say, the more you learn about the neuroscience of this stuff, I suspect the less likely you are ever to take it. I know one of the reasons my wife, who's a chemical engineer and works in pharmaceuticals, one of the reasons she says she would never take this stuff is because she doesn't trust the quality control, right? You're going to really buy a white powder from some guy making it in a meth lab in a dodgy motel? Yeah, probably not. From my perspective, am I really going to take some white powder that I know is going to kill my brain cells and cause me to, have these nasty associations and pain of withdrawal? Yeah, probably not. Likewise, if you know about cocaine, one of the things you see is it causes your blood vessels to burst. I'm not making that up. That's literally what it does. In the brain, the increase in blood pressure and the increase in hyperactivity that you see as a result of cocaine is you end up with these, little bursts, these little microvessel damage that cause you to have mini strokes, basically. And so you can track this from baseline through several doses of cocaine. All these little colored markers are areas where there are microbleeds. And so one of the many reasons why cocaine is pretty nasty is that it does cause these changes in blood pressure in the brain and so on. Many of these, even if you don't get a burst, the brain is adapting to close off the blood vessel and reducing blood flow to a particular area. And so once again, the brain's adapting and trying to protect itself. That may well cause you to have inability to use certain brain regions. So you get vascular damage, you get strokes, you get all kinds of nasty things. This also happens in the periphery. You can get heart attacks pretty easily from cocaine. It's worth noting that in some cases, the effects of a drug that we would describe as being negative or addictive can be thought of as self-medication. If you look at the volume of gray matter in the healthy brain, one thing that you see in folks who are schizophrenic is that's reduced. It looks very much like alcoholic patients. And so it's quite possible that there's an underlying brain condition that causes both schizophrenics and other alcoholics to go seek out that drug. If you are schizophrenic, your likelihood of becoming an alcoholic is much higher than baseline. And what you see is, in fact, you further damage your brain matter. What the underlying causes are is not always totally clear. But it's certainly the case that for alcohol in particular, but also in some cases for things like smokers, they are trying to use this drug as a therapy for some other, often more psychological condition, whether it's depression or dealing with stress or whatever it is. There's a reason that a whole bunch of ER doctors are smokers. They know the health risks, they know it's bad for them, but it's a tool they use to cope with the repeated stress. And so any underlying mental health issue may well predispose you to be an addict of various types. Speaking of stress, rats are group animals, right? They like to be in company, they like to have other rats in their cages. If you give a random rat that's in one of those group cages access to cocaine, for fairly moderate doses of cocaine, even up to 0.5 mg per kg, basically the rat will mostly ignore it. They don't need that cocaine, it's not doing anything for them. They feel perfectly happy, they have friends. It's only when you give them a really high dose of cocaine, they'll say, okay, fine, give me the pleasure, man. On the other hand, if you take a rat and put it by itself, that stresses out the animal. It really doesn't like that. And what you can see is that at much lower doses of available cocaine, they will start really chowing down. They will self-administer cocaine much more readily. The clear implication, and this turns out to be true in humans too, is that animals who are stressed, who have other negative environmental factors going on, are much more susceptible to addiction. And I think probably most of you have seen that in humans too. Folks who are struggling with 5 jobs and they are in poverty and they have sick kids, whatever it is, much less resistant to starting to smoke or drink or take some other behavior that will at least give them a little bit of pleasure and allow their brain to maybe even get away for a while. One last example of comorbidity type stuff. You can actually do a pretty decent job of predicting someone's chances of getting schizophrenia by looking to see whether they're a smoker as a kid. So if they're smoking more than 10 cigarettes per day by the time they're 18, the chance of becoming schizophrenic is almost 3 times as high as someone who's not smoking at all. This isn't because smoking causes schizophrenia. It's because the people who have a brain set up to be predisposed to schizophrenia are also the ones who are more likely to try and self-medicate by starting to smoke, and their brain's responding to that. So a lot of these conditions have multiple underlying causes in common. How do we know a lot of this stuff? A bunch of the work in humans comes from work done in federal hospitals in the US. Not hospitals where you or I might go, hospitals associated with incarceration, with prisons. And so inmates were involuntarily and utterly unethically exposed to various kinds of drugs and made addicted, and basically experimented upon to see what was going on. These days we use rats instead, right? So here's a rat in what's called a Skinner box. You should recall Skinner, the guy that trained the pigeons. And so you have a lever, you have some light, you have lots of signals that can say, hey, do something, learn something. And in particular for drug work, you hook the rat up to a catheter that can deliver drug X into that medial forebrain bundle and teach them, hey, if I press the lever when the red light comes on, I'll get some cocaine. And so you can look at how they respond to that. This can be done in various species. You can give monkeys access to cocaine, and you see the same thing that you would see in the rat study. Dominant monkeys, the monkeys on top of their tribe, pretty low stress, resistant to self-administering cocaine until you get to pretty high doses. The subordinate monkeys, the ones that are really trying to climb to the top of that hierarchy in the tribe and are being beaten up by the dominant monkeys, much more stressed, much more likely to start taking cocaine when it's available. So we know a lot from animal studies about the link between stress and drug take. How do we intervene? This is where we start to get back to self-control. So a lot of the things we've been talking about recently, you should start to see the links between them, right? What is self-control? Well, in many ways, it's your frontal lobe telling the rest of the brain not to do stuff. In this case, it's telling your memory and your medial forebrain bundle, yeah, we know X would feel good. It would feel good to get drunk or get high or go gambling. Don't do that. And so a lot of the interventions are built around trying to increase self-control. What is self-control? What's the self? If there's something in your head that is trying to control the rest of you, is that you? Is that part of your personality? How does this work? Yeah, we get into philosophy, and we'll come back to this at the very end of the semester. But I really like this particular quote. If, in short, there's a community of computers living in my head, there better be someone who's in charge. And by God, it better be me. So the idea is that there really are different parts of your brain, often in conflict with one another, and you'd really like there to be a chief executive area, an overarching, brain piece that is trying to make decisions and trying to control what the rest of you do. You really might think about how this resembles attention, right? Something is telling your brain what to pay attention to, and the rest of the stuff often gets ignored, even though that information is at least somewhere in your brain. Something is trying to tell your brain, no, make better choices. Don't take those drugs, go to class, get your A's. What is that thing? What is that creation of self someplace? What is you? And if you start thinking about that too much, you'll end up before drinking. It's a really hard issue to grapple with. Where are you in your brain? Is there a difference between you and your brain? What is self-control? Which part of your brain is exercising that self-control, and so on? How does that change if you are a carpenter ant and a fungus has invaded your brain? Do you no longer exist? Or are you now the fungus? Or what's going on? What we're talking about self-control though, there's a really famous experiment, and it's one that I like a lot. The marshmallow experiment. This is something that, if you happen to have a parent who's a psychologist, certainly you've probably experienced this. My children have certainly been subjects in my own version of this experiment. And the basic concept is fairly similar and fairly simple and well known. You take a child, various ages, put them in a room and say, hey, here's the deal, right? Here's a really tasty marshmallow. If you can just hang out for 10 minutes, I got something else I got to go do. But you can just hang out and not eat the marshmallow for 10 minutes, when I come back, I'll give you a second marshmallow. You get 2 marshmallows. If you want to eat marshmallow now, you can do that, but you won't get another one. If you can hang out and control this for 10 minutes, I'll give you a second marshmallow. And so, there are videos of how children try to do this, and they're really quite entertaining. We'll watch one now. And don't worry, the sound is not particularly loud. No, not so good. That kid's gonna have a C- average. All right, I'm joking a bit, and it's a funny video. Bear in mind, if you take the kids in this study who resist the marshmallow and compare them to the kids who eat the marshmallow, there's more than a 200-point difference in their SAT scores. The people who are able to resist do much, much better. It's a much better predictor than, for instance, IQ. There are lots of coping mechanisms you can see here, right? Self-distraction, playing games, trying to keep yourself occupied. One thing you should realize is most of these kids are focused on the marshmallow. A very easy way to try to avoid that would be to push the marshmallow to one side, to distract yourself. Of course, the experiment is set up to avoid them having an easy distraction. They're in a pretty boring room, there's no books, there's no nothing like that. But this stuff is trainable. You can teach people to have better self-control and to learn to generate internal distractions. So this little girl, trying to whistle, not look at the marshmallow, avoid it. That's not a good level of self-control. And in fact, that particular child ended up in therapy. I'm not kidding. All right, so here's a success. The experiment is coming back and saying, look, here's the chocolate syrup I promised you to go with your marshmallow. Oh, and by the way, yeah, second marshmallow, right. Hang out another 10 minutes and I'll bring you a second marshmallow. This kid, going to Harvard, right. Now you've got chocolate syrup. It's even harder to resist. Also, wiping on your clothes, yeah. Playing games, very important to keep yourself distracted. Even if things you do to keep yourself distracted are kind of gross. Okay, here's a smart kid. Push that marshmallow out of the way. Don't look at it. Out of sight really is out of mind. This works very effectively. It's just like, avoiding the places you used to take cocaine. If you don't have that memory, if your attention isn't drawn thinking about the cocaine, you're much better able to resist temptation. Success. He didn't even touch the chocolate. He got one more marshmallow, and here's his reward. Second marshmallow, chocolate sandwich. Yay! All right, so it's a cute video. It has a serious point, right? We see strong effects of this task, even 30 years later. And so here's that original study. It's a very famous study. Walter Mischel, got a whole career out of this. So the nature of adolescent competencies predicted by preschool delay. They tested these kids when they were 4. They came back to look when they were 16, 17, 18. And what you see is that folks who managed to resist were much more what they call academic competent. They got better grades, they had more friends, they were able to cope with issues in their lives. All of these things were well predicted by their performance on the marshmallow test, just being able to resist a marshmallow. And these are pretty big effect sizes, right? The marshmallow test has a lot of predictive power. If you come back 30 years later and test those original subjects, so this is the same group, track them down 30 years later, what they found was differences in body mass. What they saw was it's not a huge effect, but folks who had managed to not eat the marshmallow, the longer time they managed to not eat the marshmallow, the thinner they were as an adult. And so BMI, things that you might expect to be predicted by self-control. Here's a really nice follow-up study. So that original study, pretty decent, well-designed. They really didn't think necessarily about whether all the kids came in with the same expectations. This is a follow-up study, 2012. So after more than 30 years later. And what these guys asked was, can we manipulate how the child thinks about the experimenter to modulate that marshmallow behavior? So what they did was try to simulate home environments where maybe if you didn't eat a marshmallow right away, if you were given a marshmallow and you left it there for 10 minutes, your big brother came along, beat you up and took your marshmallow. And so it was rational to eat the marshmallow right away, even at the cost of potentially having 2 marshmallows later, because more likely you get no marshmallows. So what these guys did was have an experimenter come in beforehand and say, hey, thanks for coming to the experiment. It's going to be about 15 minutes. Would you like some drawing materials? Would you like to do some coloring? All the kids basically said yes. And then the experimenter either came back with drawing materials or just didn't follow through. So they had this condition where they'd either set up the experimenter to be trustworthy or not trustworthy. And here's what you see. If the experimenter had proven reliable, now more than half, 60% of the kids could wait 15 minutes. That's a huge increase. Conversely, if the experimenter didn't come back with the drawing materials, and now they say, hey, if you wait 10 minutes, I'll bring you another marshmallow. Well, why are you going to believe them? And that's actually what they saw. Less than 10%. Fewer than 1 in 10 people of these kids in the study where the experimenter hadn't brought them the crayons, now was able to resist and wait for the marshmallow. And it's rational. These kids had learned, no, the experimenter's lying. I'm not going to get a second marshmallow. I may as well eat this one. These kids had learned, well, she followed through last time. I guess I believe her. I'll hang out. And so I really like this as an explanation for some of the variance. It's not just that individuals' brains are differently wired and that you can't control whether you'll get fat later in life and whether you'll have better SAT scores. A lot of that is actually driven by your home environment. If you have a home environment that rewards delayed gratification, that rewards putting the effort in and waiting and getting your reward later on, well, that's going to allow you to, when faced with a challenge like this, make the decision. Okay, yep, I'll wait. I'll have 2 marshmallows later. This is just like going to class and studying. Every morning, well, until we were doing this asynchronously, you had the choice. Okay, I can get up, or I can stay in bed. It's nice here and cozy. The getting up kids, these are the ones, that are waiting and getting the second marshmallow. They're going to put in the effort now. They're going to resist the single marshmallow, the stay in bed, for the reward of the double marshmallow. They get an A at the end of the class. And so that kind of behavior comes throughout our entire lives. The ability to control your impulses and your short-term gain for long-term reward. Once again, this should remind you of a previous comment. This should remind you of anorexia. Basically, there's a healthy balance here of being willing to control your short-term impulses for long-term gain. As you might expect, it's possible to go too far. It's possible to never allow yourself to have any short-term pleasure because you perceive it as being guilty, or you feel as though you should just put it off and wait for something better in the long run. There's a balance. You want a healthy balance between those short-term and long-term stuff. But this experiment where they manipulated how the child felt about the experimenter, I think that's a really neat experiment to look at a previous, very famous set of work and say, yeah, but what about this? Did they think about this other angle on the work? All right. Biochemically, what is self-control? Yes, some of it is frontal lobe activity. What you also see is in this nucleus accumbens, in this reward pleasure pathway, the number of dopamine receptors actually can predict some of these self-control aspects. So impulsivity, the ability to give in to a stimulus as opposed to wait. So this is work done in rats. They first of all took all the rats. They hooked them up to a lever that gave them a cocaine. But it could only do so after a certain amount of time had passed, right? So you had to wait, I forget, 5 seconds maybe, before being able to press the lever again to get more reward. And if you pressed it too soon, you reset the clock. And what they saw was that rats spontaneously differed. So days 1, 2, 4, and 5, there's no particular reward here. Day 3 is the cocaine day. And what you can see is that some rats make many more premature responses than others. They're less able, you might say, to control themselves from pressing the lever too early. And so these were assumed to be high impulsivity rats compared to the group down here that only pressed the lever too early about half the time, low impulsivity rats. If you look in the brain of those rats, what you see is that in the striatum, in the ventral striatum in particular, not the motor area, but the reward pathway, in the high impulsivity animals, there is less dopamine binding. How do we want to think about this? If we assume that dopamine binding corresponds to pleasure, in the high impulsivity animals, they're getting less pleasure from a particular stimulus. So they press the lever, they get the cocaine, they get a buzz, but not as big a buzz. And so they're really anxious to press that lever again and get more of a buzz. The low impulsivity folks, on average, getting more cocaine binding when they press the lever the first time, better able to control their desire to press the lever again until it's the right time. All right, that's the theory. If you then give these animals free access to cocaine, what you see is the dark circles are the high impulsivity animals. And over 20 sessions, they consistently give themselves more cocaine. This is entirely in line with our theory that they're trying to achieve basically homeostasis. If they're getting less dopamine binding for every lever press, then they're going to press the lever more to achieve the same total amount of cocaine as the low impulsivity guys are doing with fewer lever presses. All right, fine. So far, so good. Here's the actual experimental payoff. So they replicated the impulsivity, same animals. They saw that the high impulsivity rats were still very high impulse, the low impulsivity rats much more. And then they gave them 3 sets of self-administering cocaine. They allowed them to give themselves as much cocaine as they wanted during that time. What you see is that entirely wiped out the difference in impulsivity. If the rats were able to pre-treat themselves with as much cocaine as they wanted, now they were no longer unable to control themselves. Now they'd actually basically self-medicated and controlled their impulsivity. This isn't just true in rats, it's true in humans. And it's not just true for cocaine, it's true for behaviors as well. Gambling is the best studied one. And so people who have gambling addictions, one of the most effective ways to control that is to give them some alternate source of pleasure that replaces the need to gamble. Another context in which you see something very similar to this is people who are trying to quit smoking. Very, very commonly gain weight. Why? Because they are seeking out an alternate source of pleasure. They've got this homeostatic need for a certain level of dopamine binding, a certain level of pleasure. And if they can't get it from nicotine, they will instead get it from something else, often food. And so you very commonly see weight gain in folks who are trying to quit smoking or quit cocaine or whatever, because they're doing this homeostatic adaptation. They're taking some other source of pleasure to make up for the missing stimulus. If you measure dopamine for cocaine, for amphetamine, you do this for nicotine, you can do it for gambling. What you see is that you get the same pattern basically. There's an initial rush, and then if you keep giving yourself the same stimulus, eventually that amount of dopamine wears off. That's tolerance. You get less dopamine binding for the same stimulus, and we'll see why in a second. It's worth noting, by the way, that, eventually this adaptation to a certain drug causes you to physically look like someone who has, for instance, Parkinson's disease. So if you're a meth addict, here's dopamine binding in the control brain, here's dopamine binding in a meth addict brain, and by now you should realize this is the brain's adaptation to defense mechanism. If you're constantly overstimulating that dopamine pathway, your brain's going to downregulate. Your brain's going to say, wait, I'm getting too much dopamine release over and over again. I'd better adapt to that by reducing my amount of dopamine binding. And so, here's a more diseased state where it happens without any external input, but you can see in both cases, you've got a loss of dopamine binding. I mentioned adaptation. That happens in the very short term too. If you have nicotine, for instance, you give a single burst, you get a certain amount of dopamine release. You give a second burst, you get dopamine release, it goes down a little, and it will continue to go down and down. The same dose will give you a smaller response, and that's because of this adaptation, because of this tolerance. If you take cocaine, the brain's going to say, holy cow, that's too much dopamine. I'm going to reduce my dopamine response. And so the next time you get a dose, you don't get as much of a response. And as you might expect, that actually causes you to increase your dose, and that way lies addiction. The opposite of addiction is withdrawal. And if you're wondering what's on the screen, this is a cold turkey. So going cold turkey is the common slang for cutting yourself off from a drug, going through immediate withdrawal from a drug. Withdrawal is the opposite of tolerance. It's the opposite of addiction. And that's true biochemically, it's true experientially. The reason that withdrawal hurts is because it's the opposite of the pleasure you were getting from the drug. And we'll see why. So what is tolerance? Defined here on the screen. You're shifting your dose response curve to the right. So you take a little cocaine, you get a buzz. Next time, because your brain's adapted, because you have this homeostasis going on, you'll get a little less buzz. So maybe you'll take a little more cocaine. And that process continues until where once you were taking, I have no idea how much cocaine one takes actually. Let's say you started out taking 1 gram, you might end up taking 100 grams to get the same amount of pleasure. A couple of things related to that. You'll tend to get cross tolerance to similar drugs. So if you're addicted to a stimulant like cocaine and you start taking meth, which is also a stimulant, you'll probably need a higher dose than someone who isn't a cocaine addict because you have that cross tolerance. The other example here, alcohol is a depressant, benzodiazepines are also a depressant. There's a reason you shouldn't take them together. They have synergistic effects. But if you're an alcoholic, it's likely you'll actually have a much smaller response to benzodiazepines. 2 other worrying things. Generally, the tolerance is most seen for that pleasure. It often doesn't apply to things like heart effects. So cocaine, the most common way you die from cocaine is you get a heart attack. And that's because you're getting tolerance to the pleasure. So you're building up the dose that you're taking. You're not getting tolerance to some of the peripheral effects, including things like elevated blood pressure and heartbeat. And so eventually your heart can't take it. And you take a dose of cocaine to get that same amount of pleasure that causes you to have a heart attack. Even worse, and again, I'm going to use cocaine as an example, you can have this inverse heart sensitization. And the phenomenon here is that you take a drug and next time you take it, some of the symptoms are worse. Paranoid schizophrenia in cocaine addicts is a good example. So what tolerance looks like in the dose response curve, here's the initial curve. You take a certain amount of drug, you get a certain amount of pleasure. Let's say you take enough drug to get this amount of pleasure. This is your initial dose, that's what you want to get. Now the second time, if you take that same dose, you'll only get this amount of pleasure. You're on the red curve. Your dose response curve has moved to the right. And so in order to get the amount of pleasure that you wanted, this amount up here, now the dose you have to take gets much larger. Tolerance is a buildup so that you need a bigger dose of drug to get the same effect. Or conversely, if you take the same amount of drug, you get a less effect. And withdrawal, it was the opposite of tolerance. So if you've been taking let's say cocaine, and it's making you get pleasure, your brain will adapt, and the tolerance says, well, okay, the new normal is I have cocaine. So at baseline, I'm kind of expecting a small amount of cocaine. Most smokers will tell you that exactly this, that after a fairly short time, it's not even that you're getting so much of a buzz from the cigarette, it's that you're avoiding the pain of not having a cigarette. And so your brain has said, okay, this is the new normal. The new normal is to smoke or to take cocaine. And so now in the conditions, when you stop taking that drug, you get symptoms that are the exact opposite. If the normal was, hey, I took some cocaine, I felt either good or at least normal. Now when I don't take cocaine, I feel really bad. I feel exactly the opposite of how I felt when I took the drug. And that's like, as it says here, the body has compensated for the drug's present. Now when it's absent, you get these withdrawal effects. And so this is the summary, if you like, of tolerance and withdrawal. As you take drug again and again and again, drug exposure, the amount of effect goes down. So let's say this is pleasure on the y-axis. You start out with a small amount of drug, making you feel really good. As you get more tolerant, that pleasure goes down, even if you take bigger and bigger doses. Then you go through withdrawal. You stop taking the drug. Well, you were at this point where with a large dose of drug, you were just above baseline pleasure. Now, when you take the drug away, all those neural changes that have adapted to the presence of the drug, they make you feel exactly the opposite. They make you feel really bad. And as you go along through withdrawal, your brain will adapt back to baseline. And so eventually you will, assuming you manage to stick the course, come back to the point where you don't feel those withdrawal symptoms. Your brain will come back to baseline. But it can be really intensely unpleasant. Something like nicotine, really hard to keep the habit because it feels so bad. It's physical pain. And so we're going to spend a little time talking about some of those individual drugs. You know, this is really not a drug class. Mitch Earlywine teaches the addictive and compulsive behaviors class, commonly known as the drug class, and we'll happily go into far more detail. But it's really relevant to many students and to family members and so on. And it's a really excellent example of how the brain does change in response to different stimuli. So the major, addictive and psychoactive ingredient in tobacco is obviously nicotine. It's extremely addictive. About 70% of folks who smoke, I really do mean even once, become hooked. And it's really hard to quit. Most of the major negative effects from smoking, let's be honest, are not from the nicotine. They're from all the other crap that's in the cigarettes. The idea of taking something into your lungs where you burn stuff. I took my children to see an exhibit called Bodies, where entire human bodies have been plasticized and different layers peeled away. All the people in the exhibit were Chinese individuals, and they were pretty much all smokers. And so their lungs were blackened because of the smoke. I think that's a pretty effective way, in fact, of convincing your children not to smoke. If you need nicotine, take some gum, take a patch. Please, if you learn anything from the class in terms of your own behavior, don't smoke. It's really bad for you. It's really harmful. It smells terrible. It's a dumb idea, right? Please don't do this. But nonetheless, it's also really hard to quit. It's only about 1 in 5 people who try to quit tobacco manage to do so. The other most common by far psychoactive drug of addiction, besides nicotine, is alcohol. And alcohol is a little weird in terms of addiction, because unlike almost all other drugs that you might get addicted to, it doesn't cause a huge dopamine rush. It's not stimulating that forebrain bundle. It's actually a depressant. It's a GABA agonist. So it's turning off different brain regions. In particular, it turns off in sequence your frontal cortex. So you reduce your inhibitions. That's chemically and psychologically absolutely true. You turn off the brake on other behaviors. This is why you jump on the table, start dancing. Then it turns off your cerebellum. Well, that's why you fall off the table, because your balance is gone. And then it turns off your hippocampus, which is the memory area, which is why you can't remember the next morning that you were dancing on the table. Like many drugs, If your parents are addicts, it's much more likely that you'll become an addict. There's definitely something genetic about the makeup of the brain and presumably the expression of, let's say, dopamine receptors or whatever, that makes you more likely to be addicted. You know, we talked about having an addictive brain type. That's both true and inheritable. You get definite tolerance. You know, people who drink 5 pints of beer, 7 pints of beer, 9 pints of beer. When I was an undergrad, some of the rugby players I played rugby with could drink 17 pints of beer and still appear to be functional. You definitely get a huge tolerance here. And it has widespread tissue effects. Ethanol can actually kill cells. If you look at the effects of ethanol in terms of brain damage, this is a brain slice from a rat, and various other drugs that cause overactivity, what you see is that the fact that ethanol is turning off some cells probably causes other cells to be hyperactive and leads to loss of cells in brain death. And certainly if you give it to a rat, the rat will overdose and die. This is two rats, both of which have alcohol. This one had an alcohol blocker, basically prevented the metabolism of alcohol and caused the rat not to drink because it felt painful. So alcohol can have pretty deadly side effects. In general, the most common likely cause of death in humans is that because it's a depressant, if you drink too much, you can go into coma and eventually die. You can drink yourself into a stupor. And the more that you get addicted, the more your tolerance builds up, the more likely you are to drink so much that happens. If alcohol is not releasing dopamine, how is it addictive? This is true for rats too. You can't actually directly addict a rat to ethanol. You have to mix it with sugar water to get the rat hooked on the sweet taste, and then they will keep self-administering ethanol. In humans, a big part of it is almost certainly the social aspect and the reduction in pain aspect. So ethanol is a GABA agonist, like I said, a depressant. It turns off brain regions. That includes turning off pain, and in certainty, turning off the ability to be aware of your pain. People who drink to get drunk often have something they're trying to escape from, whether it's physical pain, mental trauma, deep unhappiness. The more pleasant flip side is that you also can become addicted to the pleasant social effects of reduced inhibition. If you're in a group of people, all of whom are somewhat tipsy, then it can be very fun. You can enjoy each other being, some people describe it as being more real, certainly feeling less constrained by your frontal cortex to not enjoy the moment. Conversely, if you're the only sober person in a bunch of drunk people, they can appear really weird and out of control and unpleasant. But there's definitely a social aspect to the addiction in humans to alcohol in many cases. You get addicted to the enjoyment, the partying, the pleasure from the behaviors rather than from direct dopamine release. I mentioned it's heritable. That's true for basically all addictive behaviors, addictive substances, whether it's gambling or smoking or LSD, less for LSD, more for cocaine, more for opiates. Caffeine, we're not going to talk about caffeine as a drug of abuse, but along with nicotine and alcohol, those are the three big psychoactive substances that many, many people consume. And just to finish with, let's have an example of specific withdrawal. So alcohol is a depressant. If you are addicted to a depressant, your brain adapts by essentially applying a stimulant the whole time to counteract the effect of the drug. So if you are a long-term high alcohol user, your brain has adapted to compensate for that, and so will be actually more active without the alcohol. It's going to increase baseline activity. So now you take the alcohol away. Well, your brain is hyperactive. That's absolutely true. You'll get these, overactive consequences, things like nausea, sweating, vomiting. You can, in many cases, if you are a severe alcohol addict, have convulsions. You have seizures. Think about this. Your brain is overactive. You've taken away this brake, this GABA agonist that you're being used to administering every day. You've taken away the alcohol. The response of your brain is to, wow, overactive firing. That's a seizure. The extreme case, delirium and tremens, called the DTs, that's basically the whole body shaking, lots of seizure activity, lots of convulsions. If you just go cold turkey withdrawal from alcohol, it's extremely debilitating, extremely painful. extremely unpleasant. If you survive, it'll last 3 or 4 days. It can definitely be fatal. In some cases, for some drugs, and alcohol is one of them, the consequences of trying to withdraw cold turkey can actually be worse and more dangerous than the drug itself. You can imagine if you have a high-level cocaine habit, you're addicted to a stimulant. Now, if you try and go cold turkey and suddenly take that away, well, your brain has become used to having a very low level of activity, putting a lot of brakes on activity. You take away the cocaine, you can go into a coma and die because you no longer have the stimulant your brain's adapted to. And of course, there are peripheral symptoms, Korsakoff's, cirrhosis. One last thing worth mentioning is that, particularly for alcohol, the consequences of having a parent who is an addict, or a mother specifically who's an addict while you're in the womb, are really quite awful. The effects on the fetal brain are devastating and irreversible and cause marked mental retardation, marked loss of brain development. It's a really bad idea. As far as we can tell, there is no safe dose of alcohol during pregnancy. Even one drink can probably have some effect on the fetus, no matter how small it is. It's a little hard to tell, but that's the best advice. On which unpleasant note, we'll end. We'll come back and do marijuana and so on next time. Again, we're not really going to do marijuana, right? We'll talk about that. And I look forward very much to seeing you all and talking with you tomorrow. Hope all is well. See you soon. Bye, guys.